came up over here, we could all stand in front of my computer. Let's see it there. Okay. Okay, that was fun. I don't know what's going on. I think I need a new computer. So someone emailed me that lecture 6 wasn't up. I did try to post it. There's something wrong with the file because I don't know if you guys remembered on Tuesday in the middle of when I was walking around talking to people during that small exercise, it said that my hard disk was full and then it went away for a minute and I think that destroyed that file. It's on my computer, but I couldn't export it, so I'm working on fixing it. Don't worry, there's about 3 years worth of cell bio lectures up on the YouTube. Can you guys? Okay, thank you. There's about 3 years of cell bio lectures up there, so when in doubt, just find an old lecture, and the bulk of the material should be there. But hopefully it'll get repaired. I downloaded a program that repairs movie files. So we'll see what we can do. I think what happens is if at any point when you're recording a movie, there's a power surge or something changes, then that file gets corrupt. And so that's what happens. Okay, so quick business. Those who haven't brought Scantrons, please remember to give them to me in recitation. Exam is coming up in about 2 weeks. And then Stump the Teacher. We have to Stump the Teacher about transport to the lysosome, right? Autophagosome to lysosome. And I very quickly set up Blackboard, and everyone submitted it there. And so that was great. If you had trouble submitting it, always email me, but I ain't getting any Connect email, so you should be good. And then it was very easy for me to just go through and grade it. So if you see a point showed up in your grade center, that means you got the point. If a point didn't show up there, that means you didn't get the point. It should be there. But you have to let me know if it comes up 0 or 1 or how it looks on your side. On my side, there's a column that says Stump the Teacher with a date and either 1 or 0, or if you didn't submit it, it doesn't exist for you. So let me know. Oh, the answer. Yes, well, we kind of knew what the answer was. The question was about snares and whether they're involved in autophagosome lysosome fusion. And we kind of assumed that they were because most all vesicular transport uses that mechanism. And so, yes, they were. And then a number of you guys gave me exact mechanism and snares. And those of you guys who gave me exact mechanism and snares got the points. Those who just said, oh, yeah, snares are involved, well, that's not the question because that's what we spoke about in class. So that's those of you who didn't receive the points is because you said snares are involved. You needed to tell me exactly which ones. And don't worry, you don't need to know which ones they are for the exam. I can't even remember. But you did tell us like syntax and something and vamp something. And I don't know. Anywho, good job. A lot of people participated in that. Okay, so we're going to take 5 minutes to write. Actually, it's more like drawing today. So we talked about transport of proteins from the Golgi to lysosome. And we spoke about that on Tuesday. So I want you to kind of take 5 minutes to draw out the process of what needs to happen. So make sure to include your protein, your lysosomal protein with the correct modification, receptor showing vesicle formation with all the components required for vesicle formation. and protein binding and the Golgi release enzyme. So there are certain things that dictate whether the protein will bind to the receptor in one place and be released in the other place. You want to include that too. And so then we'll go over it. Okay. So, does anyone feel really good about what they do and want to come up and share their drawing? Uh-oh. Or how about this? Does anyone feel really good about what they drew and want to quickly go over it with me in a quiet way, and then I'll take their drawing and go over it? No. People don't like to stand up in front of people. I get that. I understand. Okay. So, before I draw it out, I want to hear what people have to say. Michelle Jurjevic?
Okay, I forgot my notes, so I kind of do it from memory. But I think it was, I have that the lysosomal protein has like a signal patch, and then the Catherine like coats where it's coming out from the plasmin membrane. I think it was.
Okay, stop right there. Okay, so I heard the keyword signal patch, which is associated with this function. And then I heard coat proteins and plasma membrane. And so that's where I'm going to stop you because we're talking Golgi to late endosome, not plasma membrane.
Okay.
And so the reason I put this in is because I want to make sure people can keep those two things separate. One is Golgi to lysosome, so that's the biosynthetic pathway. Yes, signal patch is a good thing to be remembering. But this is not endocytosis.
Okay.
So, and there's a few people I saw doing the endocytosis when I was walking around. Yes.
I did the lysosomal protein with the M6P signal patch binding to an M6P receptor in the Golgi membrane and then letting off with the help of clathrin to form a vesicle, which becomes the endosome, and then as it becomes more acidic, it turns into the late endosome. And then the only thing I wasn't sure about is if it actually, that endosome fuses with the lysosome or if it's somehow.
It's more of a maturation process. It's not completely clear in the book. So just think of late endosome lysosome so the question was really about is the late endosome fusing with the lysosome? Is it becoming the lysosome? Is it the lysosome? And so just think of it as it's going to the late endosome, and as long as there's low pH and you have active acid hydrolysis, it's functioning as a lysosome, right? But you're all right in what you're saying. Okay, so let me just quickly trying to keep track of who I call on. Okay, so that's good. So right now we've got lysosomal protein. It has a signal patch. So it's recognized. One thing you guys both left out was it's recognized by an enzyme that binds to the signal patch and that transfers mannose-6-phosphate. So that's the correct modification is mannose-6-phosphate. The correct receptor is mannose-6-phosphate receptor. So this is all happening in the Golgi. There's a mannose-6-phosphate receptor that's going to bind to that lysosomal protein by interacting with mannose-6-phosphate. And then, you get clathrin and vesicle formation, absolutely. Goes to the late endosome. And so then I said mechanism for protein binding in the Golgi and release in the late endosome. Who had that part of it? So there's got to be something that's going to regulate where the lysosomal protein is picked up and where it's dropped off, right? You don't want to be picking up lysosomal proteins in the lysosome and bringing them to the Golgi, do you? No, right? This mannose-6-phosphate receptor is shuttling back and forth. Yeah. The pH, right. So remember there's that theme of pH gradients controlling proteins binding to receptors and moving things around. Yeah.
So it's just the mannose-6-phosphate?
Mannose-6-phosphate is on your lysosomal protein. Mannose-6-phosphate receptor goes back and forth. Yeah.
It's like a shuttle.
It's kind of a shuttle mechanism, exactly. Yes.
What was the enzyme?
Mannose-7-phosphodiesterase, something
And
phosphodiesterase.
what does it do?
It's adding mannose-6-phosphate to the lysosomal protein. Yeah.
So does the early endosome have acid hydrolysis in the vesicle?
Early endosome, I believe may or may not have acid hydrolases, but the pH isn't low enough for them to be very active.
Does it have like a proton pumping?
It does have a proton pump. It's a lower pH than, say, the cell surface or the cytosol, but it's not as low as the lysosome. So going the maturation and from early endosome, which by the way has to do with endocytosis more than this process, but there's a difference. The maturation basically is a result of decreasing pH due to increasing activity of the proton pump. Okay. So let me quickly draw this out, and you guys can tell me if you got the same thing. Huh, that's interesting. Okay. Oh, there it is. Huh, weird. Okay. So you would have to have a cell. We're just going to do, here, we'll do the whole cell just for fun. And then, here's your nucleus. And then here's your ER. Obviously, it doesn't really look like that. Here's your Golgi. Obviously, it doesn't really look like that. And then we'll call this lysosome for the sake of simplicity, right? And so the process we're drawing is Golgi to lysosome, right? And so in here, that means you have a protein, and it has a signal patch, and it's going to interact with this something phosphodiesterase or something like that. I don't remember what it's called. What's it called? Phosphotransferase. Duh, it's not cleaving. Thank you. Phosphotransferase. I'm not very awake this morning. You'll have to excuse me. Who feels groggy this morning? I think it's the weather, right? Because I was not out partying last night, just so you know. And I feel like I was. Okay. Ugh. Transferase, see? Okay, so phosphotransferase. What's the phosphotransferase do? Right. So it's going to add that mannose-6 phosphate. So M6P. Okay. And so then that protein, you're going to have your M6P receptor, right? And that's going to bind to your protein. I should have made this bigger with your M6P. We'll just make it a big black dot or blue dot. And then you're going to have clathrin, right? That's all going to form. And you're going to have your vesicle budding. That's going to have the receptor containing your protein with your mannose-6P, right? And that's all dependent upon the pH being high enough so that binding can happen. So I'll say higher pH. I wouldn't call it a high pH. It's a higher pH, and this would be your lower. And so it's going to go here, and then it's going to fuse, right? And that's all still based on your mannose-6 phosphate receptor, but at the lower pH, it's going to be released. So now it's in here. Release of hydrolase. And so then that vesicle another vesicle could bud, right? That's going to bring mannose-6 phosphate receptor back to pick up more acid hydrolases. Hopefully you followed my groggy drawing. Yes?
Is the colored in dot?
That's a mannose-6 phosphate.
That's
Yeah, so I just, because I realized I was writing everything too small, and I couldn't fit mannose-6 phosphate. It looks big up there, but it's actually very small in here. Yep, color dot is mannose-6 phosphate. It's all dependent on that interaction. Okay, I'm going to move ahead because, well, it's time. Okay. Okay, so this is what we've been doing so far. All the secretory pathway stuff, right? Yeah, on Tuesday we started talking about sending proteins through the secretory pathway to the lysosome, and that's what we just drew out and talked about. That's that mechanism. That's the level of detail I want. And then we started and so we also kind of started talking about how there are different ways to get to the lysosome, either through endocytosis or through the secretory pathway. And I was calling the secretory pathway a biosynthetic pathway, which was confusing people, but I thought it would be helpful because then you would be able to think in your mind biosynthesis. When I'm making things, and it's a protein that needs to go to the lysosome, it follows that pathway, Golgi lysosome. Endocytosis is bringing in things from outside of the cell. But anywho, it's good to kind of think about the different types of proteins that would go to the lysosome via secretory pathway versus the types of proteins that would go to the lysosome via the endocytic pathway, and make sure you really keep those separate, okay? And then we were talking about how proteins in the secretory pathway get diverted to the lysosome, what we just drew. And of course, the default pathway, if not diverted, is where? Outside the cell, secretion, right. And then just for your information, the lysosome is not always the end of the line. You can get lysosomal secretion, so if things are not digestible, they'll be secreted out of the cell. This usually is more cell under stress. Also, some cells will use this process normally, such as melanocytes, and they store their pigment in lysosomes, which are called melanosomes. That's what gives our skin color. And then they release the pigment into extracellular space, which is then taken up by keratinocytes and gives our skin color. And albinism is actually a defect in melanosome exocytosis, so that secretion of pigment. Okay, so this is a nice cartoon talking, just kind of to introduce endocytosis to you guys. So it says, after years of intensive research, we finally have a clear picture of insulin action. There's insulin, the receptor, something, and its effects. That's kind of how research feels sometimes, like this much, and then there's a big black box of stuff we don't know, and then some effects, and we're trying to connect those two. Hopefully, when I'm teaching to you, I'll be teaching you full mechanisms that you can kind of learn and understand, and you won't write on the exam if something happens. Yeah. So this is endocytosis in cartoon form, right? So here it's kind of showing a receptor with maybe a ligand binding to it, and endocytosis of the receptor into the black box, and then going to that fusing with the lysosome, where everything dies a horrible death. Okay, so endocytosis. So that's the other way to get to the lysosome, right? From the cell surface inward to the lysosome. This is not the biosynthetic pathway, right? This is not the way acid hydrolases go from the Golgi to the lysosome. This is receptors bringing something in, or receptors themselves coming in to the cell through endocytosis. We also spoke the other day about different ways things get into the cell from different forms of endocytosis, right? There's phagocytosis. I want you guys to notice one thing about phagocytosis. So this is showing a cell, a white blood cell, that's phagocytizing bacteria. And so notice, here's your bacteria, these two bacteria actually. And you can see there's a pseudopod forming around the bacteria. And so I just want you guys to remember that, because soon we're going to be talking about the cytoskeleton, and we'll be talking about actin polymerization and how it affects the shape of the cell and what the cell is doing. And so I just want you guys to say, oh yeah, this is what she was talking about back then, connecting two different things in cell biology. That's because it's all connected. So that's one way. This is phagocytosis, where your phagosome is going to fuse with your lysosome and digest the bacteria. That's a form of endocytosis. And then you can have endocytosis using clathrin-coated vesicles, which is what we've been talking about a whole lot. This is showing uptake of lipoprotein into a hen oocyte to form a yolk. Here's your invagination, your clathrin. This is your lipoprotein bound to receptor, endocytosis. There's your vesicle formation. Then you can also get endocytosis using caveolae, using caveolin. Caveolae, these are pretty much just membrane domains that have a different composition, and that's what causes their endocytosis. So it's not a typical coat protein like what we've talked about with CUP1, CUP2, clathrin. It's different. These membrane domains have a lot of these GPI-linked proteins that we spoke about. Glycosphingolipids, cholesterol, and caveolin proteins, and they have a different structure. So if you compare, clathrin has more of this basket kind of shape, and caveolae have this more like wound ball of string kind of structure. Just understand there are different ways to get into the cell, different coats, endocytosis, right? Viruses like to use endocytosis to get into the cell. SV40 and papillomavirus enter the host cell in vesicles derived from caveolae. Okay, so I just want to, I know sometimes it feels like we're kind of getting into these details and we forget where we're coming from. A few people have approached me and said, oh, I don't know how I'm going to study for this. I'm feeling really lost, like there's all these details and I don't know how to organize it. And I said to them, draw the biggest picture you possibly can and go through your lectures and put each piece into the picture. Some people aren't visual learners. Then you might want to list major processes and organize everything based on what goes with what process. But just to kind of remember where we are here, remember we've been talking about transport of proteins, right? And this all goes back to, you know, you have gene expression and transcription, and then translation has to happen, and the protein needs to go where it needs to go. And so you're going to have, in what we've spoken about so far, we're going to have proteins going to the ER and through the secretory pathway, which lands in either the plasma membrane or lysosome or secreted from the cell. And then there are other ways. Next week we'll be talking about import into the mitochondria and import into the nucleus. So whenever you guys get lost, always try to back up to the big picture. Okay, so to move on with endocytosis, talk about receptor-mediated endocytosis. Right, so receptor-mediated endocytosis is very specific, right? It's not this kind of random uptake of materials from out of the cell. So usually you'll have receptor, and then the ligand will bind to it, and then you'll get endocytosis. Here it's showing clathrin. This is the LDL receptor. It's just an example of receptor-mediated endocytosis. And then you'll get endocytosis, and then you get the early endosome, late endosome, and as you move towards your late endosome and the pH goes down, then you get that, you'll get dissociation of your LDL, and your, you'll get dissociation of your LDL from the receptor, and then here's your recycling endosome. Those receptors will be recycled back to the plasma membrane so they can be used again. And then your late endosome will mature into or fuse with the lysosome and then digest the LDL particle into its components so it can be used by the cell. So why would you want, what's the difference between receptor-mediated endocytosis and pinocytosis or other sorts of non-specific, non-receptor-mediated endocytosis? What's the difference?
So
Yes?
it's
Right, there's specificity of what's being taken up. In this case, the cell's bringing in LDL. It needs cholesterol, and it needs fatty acids, and it needs these different components that are coming in through diet, and they're in the blood, and they need to be taken out of the blood into the cells, right? Mm-hmm. Yes. So usually it's, in this case, it's because the cell needs to bring something in. Sometimes there are other reasons, but it's usually specific uptake of receptor plus ligand or receptor alone or something like that. So as you notice, you have your receptor interacting with clathrin through adaptin, but it's interacting with clathrin. So what would happen in the case of the LDL receptor if there were a mutation in the gene encoding clathrin? What would happen? There'd be no vesicle formation. What would happen in terms of your LDL and your cholesterol? It would build up. Would you have high cholesterol levels in your blood? I see all of these really like what? Should we just cancel class? I feel like that'd be a faux pas on my part. Right, so if you don't take the cholesterol out of your blood by bringing it into the cell, where is it? In your blood, right? Yeah, very good. Okay. Right, so you have to think that there's, yeah. So what if there were a mutation in the genes encoding the proton pump in the late endosome? Hydrolase would not be active. What else, though? What's happening here in the late endosome? That's when you're recycling. That's when you're recycling your receptors to the cell surface, and you're separating. So basically, what would happen is you wouldn't have your LDL particle dissociate from your receptor, and then it would go back up to the cell surface. Right?
Is the proton pump a protein? Like, what is it from?
Okay, I have a question. What is a proton pump? What is it made of? Yes.
Isn't it like a beta protein? It's like a barrel shape.
Yep. It's a protein that has a structure made of beta sheets that is barrel shaped and inserted into the membrane. Yep. And its structure gives it the function of being able to pump protons into this compartment. Yep.
Are there different genes for like different clathrin, like specific vesicles? Like for this one, talking about that certain vesicle, I guess it's somewhat different. Like an LDL vesicle is different than another protein vesicle, right? So then if you had a mutation with clathrin, would it affect all the vesicles or just that one?
So the question is, if you had a mutation in clathrin, would it affect all vesicle formation or just one? It would affect all vesicle formation. The similar thing is there's something in common with receptors, and we're going to talk about that in just a minute. Your cytoplasmic tail of your receptor has a common sequence that's going to allow you to interact with adaptin, and that's why you're going to have different things. That's why you can have different receptors from it. Yeah.
So if the LDL doesn't dissociate, then it's just leaving the cell again? Is that what you're saying?
Yeah. If the LDL doesn't dissociate, it'll probably be recycled back up, I'm guessing. Because the receptors recycle back up. So it's still be down to the receptor. Okay. So, right, so this is what we were just talking about. What if the LDL receptor has a mutation that results in a premature stop codon in the cytoplasmic domain? So I want you guys to start thinking about it as in, okay, cytoplasmic domain. Well, that must be this part, right? That must be, let's see. Right, that's your cytoplasmic domain. If you have a premature stop codon, what's it not going to be able to interact with? Huh? Someone raise your hand. Someone who feels confident. Yes?
The adaptin?
Right. And if this isn't here, then it can't bind to adaptin. If it can't bind to adaptin, it can't bind to clathrin. If it can't bind to clathrin, there's no endocytosis, right? So I want you to start thinking that way. So clinical implications of a defective LDL receptor, what do you think? Yep. Yeah, it could predispose you to having high cholesterol because you have a hard time taking it into yourself. Actually, I've had high cholesterol since I was like 2. I don't know, whatever age I start testing for cholesterol. And they would tell my parents, I don't know what's wrong with my LDL receptor, I haven't checked it out. But they would tell my parents, don't let her eat bacon. And they'd be like, we're kosher. They're like, don't let her eat lots of eggs. She doesn't eat anything. I was like the scrawniest kid ever, but my cholesterol was like 250. It still is. It never changed. I don't know. What are you going to do? Okay, so endocytosis. So this is the paper we're doing. Not this exactly, but it's pretty much the paper that we're doing this week. So sometimes endocytosis can be used, sometimes it's used because we need to bring in LDL into the cell. This is a normal cellular function. But also viruses like to manipulate cellular processes. And you always think to yourself, oh yeah, they use the host transcription, translation, DNA replication machinery. They use much more than that. And so one example is HIV-NEF will downregulate CD4 by inducing endocytosis. And as those of you who've had recitation, those of you who are going to have it today, as you read in the paper, this is useful for the virus, right? Because the virus does not want to alert the immune response, the immune system, right? And so by taking the CD4 off the cell surface, it does 2 things. One is that it's not going to be able to interact with T cells. Not T cells. Not T cells. Come on, guys, help me out here. These are T cells. It's not going to be able to interact with antigen-presenting cells, and it's not going to be able to be infected by more HIV virus, right? Because CD4 is the receptor for HIV, and so by pulling it off the plasma membrane, it will prevent superinfection, which I always wondered why that was, but I believe it has something to do with its vasculitis. I don't know. So that's just one way. Viruses manipulate the cellular processes. I'm going to try to put those in as they come up because I think that makes things pretty cool. When you're learning about a basic cellular process, it's kind of cool to learn about how viruses mess with them, even though this is not a virology class. So I had the question before about, are all clathrins the same? Are all vesicles that are endocytosis the same? And I think what you're getting at is how are different receptors endocytosis, right? What's the common feature? And so you should always think to yourself this way, because a lot of different receptors come in through receptor-mediated endocytosis. And you have to think, well, what's the common thing that allows different receptors to come in that way? And so basically, there's a signal sequence. This is another theme, right? You're always having signal sequences that are directing things places. And here, there's a signal sequence that determines recruitment of these receptors into clathrin-coated pits for endocytosis. And it mediates interaction with adaptin. And so you can imagine 5 different receptors might all have this common sequence that allows interaction with adaptin. And it's called the endocytosis signal. And it has this motif Y, XX, and then this thing, which means hydrophobic. XX is any polar amino acid. And this pitchfork is any hydrophobic amino acid. And that's going to allow interaction with adaptin. LDL receptor just happens to have a different sequence, so it doesn't follow that exocytosis signal, but it still interacts with adaptin. So it's NPDY. So based on this knowledge, you should understand that many different receptors can accumulate in the clathrin-coated pit and understand why that is. Okay? Yeah.
So for the vesicle, inside the vesicle is all the same proteins are a mixture of different proteins that I see.
So the question was, inside the vesicle, is it all the same protein? Or is it a mixture of different? It can be a mixture of different proteins, right? Because based on the fact that all you need is the signal to interact with adaptin and clathrin, that means any receptor that needs to be endocytosed, as long as it has that sequence, it can go into a vesicle. Okay?
So wouldn't that actually contradict what you said? Because if that's the normal sequence for LDL, it has a different sequence, so that means it would have a specific pathway to recognize its particular sequence.
Yeah, so you're saying, oh, if it has a different sequence, does it have a different adaptive protein? It might have a different adaptive protein. Clathrin is the same. But my guess is I'm not 100% sure if there are multiple different adaptive proteins. It seems logical, and then as long as they interact with clathrin, it should be fine. But by having so the take-home message is that there's a sequence in the cytoplasmic tail that allows for interaction with adaptin, and that's what allows for accumulation in clathrin-coated pits. Okay? Other questions? Yeah,
So basically, the sequence is for the adaptive, not for.
it's for interaction with adaptin because clathrin interacts with adaptin. And that's when adaptin is an adaptive protein, and its function is kind of to be a bridge between that cytoplasmic tail or a cargo receptor tail. Remember that adaptin and clathrin are not only used in endocytosis. It's that bridge between clathrin and the receptor tail. Okay. Okay, so you have to say to yourself, wait a minute, so if they all have this sequence, why isn't everything always endocytosed? How is this regulated? You always have to ask yourself, when I tell you about something, your question in your mind should always be, how is this regulated, right? Maybe? You're like, just tell me what you know. Don't make me think. Okay, I don't want to think either. That's why I'm just going to send you all home in about 25 minutes. Except for those of you who are coming to recitation. You have to put up with me for a few more hours. Okay, so this is regulated, right? I think receptors only go through endocytosis when they need to. So this is regulated by monoubiquitination. Oh my God, ubiquitin. So remember before I was saying to you, endocytosis, this is not degradation of proteins via the proteasome. It's not. But, and I was saying, but, there's always a but in science, but monoubiquitin does signal endocytosis. Monoubiquitin. And so monoubiquitination of a receptor cytoplasmic tail regulates recruitment into clathrin-coated pits and regulates endocytosis. And so that's just something to acknowledge. Otherwise, you would ask yourself, so what does LDL receptor not LDL, but do receptors always just get endocytosed? Just randomly? No, that makes no sense. Some of them do kind of regularly transit, but some of them need to only be endocytosed when they need to, such as if you need to downregulate a receptor. Let's say you have a lot of signaling happening and you want to turn off that signaling. One way to turn off the signaling is to ubiquitinate the receptor, endocytosis, and then send that receptor to the lysosome, right? And we're going to do a paper on that. Okay. Let's do a quick practice problem to wake each other up. So you're studying a new receptor. You are trying to determine whether it participates in receptor-mediated endocytosis through clathrin-coated pits. You know the protein to amino acid sequence. How can you use your recombinant DNA technology, cloning, to address these research questions. You might say, to me, what's the question? The question is, does this protein participate in receptor-mediated endocytosis through a clathrin-dependent mechanism? Okay? So look at this, think about it, talk to your neighbor. I'll circulate to clarify if need be. I'll just leave in the corner.
Sure.
Okay. Who wants to tell me what they think? Yes.
Would you, because I remember last class we were talking about just taking off the amino acid sequence. So would you take off the amino acid sequence that codes for the adaptin, and then if it can proceed with endocytosis, it isn't class independent, but if it can't go through because it can't bind with the actin, which then can't bind with the clathrin, you don't really class independent.
So you're on the right track. I'm going to fix some of your language.
Okay.
Because sometimes it's all in the words you use. That can be right or wrong, but I know you're on the right track. Yes, so there's that sequence that we were talking about. That's that endocytosis sequence. And so you would look for it, and then you would cut off that part of the sequence. So you can't call it adaptive because then that's assuming that you're cutting off adaptin.
Okay.
So that sequence, I just got a question up here and suggestion like, well, can I make a point mutation in this endocytosis signal? And it said, yes, that's perfect. And it said, well, but will that have anything to do with interaction with adaptin? I'm like, no, that's what the signal does. That signal is interaction with adaptin. And so if you mutate that signal and then you look to see if the protein comes into endocytosis, then you know that that sequence is responsible for endocytosis. If you still see endocytosis, then it would say, okay, well, that sequence is not responsible. It must be coming in some other way. Obviously, you need to have a control. Questions about that? Good. Okay, so unless cargo is diverted elsewhere, the default pathway for molecules taken in through endocytosis is the lysosome. I want you to solidify that in your head because in recitation, Tuesday when people were drawing, a lot of people were doing endocytosis to the Golgi. And I think that's because last week's paper confused people. And so I just want to solidify the fact that ricin follows this retrograde transport pathway. That's ricin, that's Chickenpox, futoxin, follow that pathway. That's not a standard pathway of endocytosis. The standard pathway of endocytosis is to the lysosome, unless you get diverted through recycling, right? So you can get recycling, and then you can also get transcytosis, which happens in polarized cells, which have a top and a bottom and a side, like endothelial cells. And that's moving from one membrane to another. But the standard path for endocytosis is degradation in the lysosome. That is the default. Okay? Oh, interesting. Well, there you go. There used to be a picture there. So the LDL receptor, it is an example, that's what we just talked about, and when I was introducing receptor-mediated endocytosis, it is an example of recycling, right? Because remember, you at the late endosome, your LDL particle is going to dissociate from your receptor, and your receptor is going to recycle. Yes?
I had to print out the slides if you wanted to use it.
That's okay. Thank you so much, but it's fine. And so LDL receptor actually makes a scheduled trip every 10 minutes or so, regardless of LDL binding. So it's that constant coming in. So if there's no LDL in the blood, then there won't be much brought in, but the receptor will still kind of keep cycling through. So what are the fates? So you have endocytosis of the LDL receptor, right? So what are the fates of the LDL containing endosomes, right? So like we already talked about, you have a recycling endosome that brings the receptor back up. But what happens to the endosome that still contains the LDL? Lysosome, exactly, right? And that's where it gets digested down to its smallest building blocks, and then can be used by the cell in biosynthetic processes. Yes.
If the LDL receptor is not being used, would it be more effective for the cells to just turn it off instead of it going through the pathway over and over again and not going in?
So the question is, if the LDL receptor is not being used, isn't it wasteful to keep going through this endocytosis recycling process? Well, I guess you have to think to yourself, what takes more energy? Endocytosis and recycling, or gene expression, transcription, translation? And what takes more time? Because you want to be able to pull that LDL out of circulation when it's present. And so by doing that constant cycling through, it can do that. Whereas if there has to be some because otherwise there needs to be a way to kind of induce transcription, translation of the receptor when it's needed, and that takes a lot longer than just recycling every 10 minutes.
I was more thinking about a post-transition modification instead of having to make it go through the pathway at the same time.
So you're thinking, can't it be modified so endocytosis doesn't happen without binding of ligand? Yeah? Maybe? I don't know. I don't know why that doesn't happen. We'll think about that tonight while we drink. No, I'm not sure. I get your point. No, I get your point. I don't have an answer, and it's not very good stuff to teacher questions, so I'm not going to give it away. Yes?
So when you say recycling, you're saying the receptor's going to the lysosome and being degraded?
No. Recycling means the receptor is going back up to the plasma membrane.
So what were you talking about the lysosome?
So remember, with LDL receptor, you're bringing in apolipoprotein B plus LDL, low density lipoprotein, right? And that's getting endocytosed, and your LDL particle is going to the lysosome and being digested into all the building blocks that can then be used by the cell for biosynthesis of other things.
Okay.
but the receptor itself buds off into a recycling endosome and cycles back up to the membrane. Okay. So here's another example of a receptor, but this is different. You know what's happening is my computer's about to die. Okay, guys, go away. This makes me so mad I can't even tell you.